Welcome everyone to today's Endocrinology Grand Rounds. We are very pleased to have Dr. Mike Merchant, who I think is well known to most of you here. But anyway, I'll introduce him. He's professor of medicine with the Department of Medicine. He has been at UofL since 2003 when he came here as a senior research assistant and has moved up the ranks to be a full professor. Uh, Currently, he is the co-leader in the Omics and Exposure Facility Core at the Center for Integrative Environmental Health Sciences. He has been co-investigator or PI in a wide range of NIH grants, currently having being a co-investigator or a PI on 9 of those NIH grants. His main focus has been in trying to identify markers for a variety of diseases, and with particular interest in kidney disease and liver disease, toxin exposures. And so he's got numerous publications and awards. Today, he'll be talking to us about diabetic kidney disease and proteomic-based markers of kidney injury, something that we will, hopefully in the years coming by, will use these in actual daily clinical practice, moving from our good old microalbuminuria to something more more specific in predicting course of disease.
Thank you, Dr. Onkashiguna. So I appreciate your being here today. I know it's late in the day. I typically have lots of slides and I talk slow, but I'll try to do better today. I'll review some basic information on diabetic kidney disease. I think you probably all are well versed in that, so I'm not going to change your thoughts on that, but I'll spend most of my time speaking about 3 different studies addressing discovery of new markers for progressive renal function decline in type 1 diabetes mainly, but also type 2. And then I'll discuss some work that I'm doing with investigators out of Pittsburgh on a similar topic. So I have nothing relevant to disclose. Topic outline has already come about, a little bit on diabetic kidney disease, a little bit on clinical proteomics, a little bit different from classical proteomics, and I hopefully you'll pick up on this when I finish with a few slides. The topic of the 3 publications or the 3 research projects, these are all out of, 2 are out of the Joslin at Harvard and 1 is out of Penn. And it's on the idea of kidney risk inflammatory signatures. And then our kidney disease degradomics, that'll be the one for our study. Some colleagues and collaborators on these projects and some funding. So my little attempt to discuss diabetic kidney disease. We have some really good markers for diabetes and diabetic kidney disease. So we have hemoglobin A1C, we have fasting plasma glucose and oral glucose tolerance tests. And these are all kind of surrogate derivative values that give us some indication on what is going on or what has transpired over the past 6 months. Right, so these are kind of the best that we have, and so everything that we do kind of will be referential back to this to see can we outperform that. Diabetes and kidney disease are number 7 and 8 on the leading causes of death. So that's the attempt to justify spending time, effort, and money studying this because it's going to have a large impact if we're able to make some progress. And the prevalence of diabetes over the next 20 years is going to approximately double. This data is kind of a global perspective, the different regions around the globe, different populations, but overall it's a steady, sustained increase. So those are kind of the driving reasons for studying this. And then in regards to chronic kidney disease and its increase with people with diabetes and length of time diagnosed with diabetes. This is a recent publication out of C. Jason. where you can see that individuals with the clear bars, these are people with no diabetes. Then you have undiagnosed diabetes and the hash marks, and then the purples are different lengths of time with diagnosis of diabetes. You can see that there's an increasing proportion of the population of individuals with albumin creatinine ratios of less than 30 mg per gram, greater than 300 mg per gram, or EGFRs above CKD stage 3 or below CKD stage 3, that the longer you have diabetes, the worse it is for you. Right. So in terms of trying to predict progression, a lot of early studies that focused on macroalbuminuria, microalbuminuria, were kind of limited in their utility because of the populations of patients that they studied were individuals who already had the disease or had complications associated with the endpoints that they wanted to study. And so there was a bias in the recruitment of individuals. And so there was an overinterpretation of the value of microalbuminuria. And another problem with microalbuminuria and proteinuria is that you can have fluctuations in the amount of that protein in the urine. So depending upon the cross-sectional day you take the urine sample, you may have a relatively high value or a small value. So things like microalbuminuria need repeat measurements. And even then, you can change year to year where you can have remission. So classically with diabetic kidney disease, there's a little bit of a snidely whiplash in the room, and that's reactive oxygen species. So the general idea is that you have a lot of sugar, a lot of glucose. Glucose gets transported into the cells, the epithelial cells, the endothelial cells. Then it starts to it's metabolized, goes into the mitochondria. They enhance metabolism, oxidation, generates reactive oxygen species. Those reactive oxygen species cause damage. You also have the polyol pathway, the hexosamine pathway, the protein kinase C pathway, and the advanced glycation end product signaling pathway. These are all getting kicked off by glucose. And we also have the problem of reactive oxygen species. So in terms of kidney disease, there have been major clinical trials addressing both glucose and reactive oxygen species. And for a while, there was a lot of hope being placed in a drug, bardoxolone methyl, to be a inducer of the NRF2 pathway, the antioxidant pathway, and its ability to correct diabetic kidney disease. And that really hasn't come through. With glucose itself, you all know well about the SGLT2 inhibitors and the outstanding job it's doing on correcting kidney disease in general, not just specific diabetic kidney disease. But reactive oxygen species tend to be the bad boy in the room. And it's been studied extensively, and it hasn't resulted in a lot of solutions on the ability to predict what's going to happen, right? So we know that there's glomerular involvement. The glomerular involvement involves glucosuria or proteinuria. We know that the glycocalyx around the endothelium is denuded. We know that there's podocyturia, so the visceral epithelial cells and the glomerular tuft are lost. We know that there's mesangial expansion, and there's TGF tubuloglomerular feedback hyperfiltration. And we know that all of these things, they've been well studied, but they haven't led to any knowledge on the predictive capacity of who's going to have accelerated loss of kidney function. Similarly, with tubulointerstitial fibrosis, these are features of diabetic kidney disease and the end organ damage that are being driven by the glucose and the reactive oxygen species, whether it's coming from effects on the bone marrow derived fibrocytes or EMT, endothelial to mesenchymal transitions, forming more of a fibroblast-like cell that's generating cytokines and chemokines, that's stimulating some differentiation and fibrosis production by the myofibroblasts. Again, all of these things are not predicting who's going to have renal function decline. So this is where a little bit of discovery science comes in, a little bit of proteomics, and specifically clinical proteomics. So proteomics is a field of study where you're trying to essentially inventory all the proteins and all the protein post-translational modifications within a given compartment. So it's either a tissue compartment, a fluid compartment, an interstitial compartment. It may be circulating and representative of the systemic system, or it may be very focal, such as in just the glomerular compartment. So clinical proteomics is a little bit different in that classical proteomic methods typically utilize a mass spectrometer. And so you're collecting a sample of your proteome, you're digesting it with a protease to convert everything into a small fragment. And then you're using the mass spectrometer to analyze those fragments. And with the mass spectrometer, we can identify the fragments, and from the relative signal of each one of the ions, we can get some sort of relative abundance factor. And we can compare sample to sample to sample. And from that information, we're able to compare relative abundances between groups of samples and infer some aspect of biology. With clinical proteomics, typically what you're trying to do is start with a clinical sample, such as a plasma sample or a serum sample. And that type of sample is problematic for proteomics in that your blood, about 50% of your blood is comprised of just albumin. About 20% of your blood is just Ig. So you can rapidly look at about 12 to 14 proteins, and about 95 to 98% of your proteome is just of those proteins. So you don't need a mass spectrometer to characterize those proteins. It's been done very nicely with antibodies for a long time. So a problem with clinical proteomics is the ability to penetrate with it through the high abundant proteome into the low abundant proteome, which is typically where all those things that people who study pathobiology are interested in. All the interleukins, all the chemokines, all the cytokines, those things are circulating in your body at a concentration of picograms per mil, right? So there have been two different methods developed for clinical proteomic study. And these are things that you'll probably end up seeing in clinical practice for a couple of reasons, and probably not for a couple of reasons. So these methods rely upon something called an aptamer. That's a nebulous word. An aptamer means a molecule that binds to stuff. In some regards, an antibody is an aptamer. So you have an antibody that recognizes an epitope. An epitope is actually about 7 to 8 amino acids. So every antibody is very specific for about 7 to 8 amino acids. The first aptamer process that came out was by a company called SomaScan. And so what SomaScan used was a organic compound that was fluorescent. And there were some combinatorial chemists that were able to have, they had such an extensive library of fluorescent molecules that they could combinatorially make millions of different unique molecules. And what they would do is they would just simply have a library that they screened for binding. And they would just look for a molecule that would bind protein A specifically, a molecule that would bind protein B specifically, D specifically, et cetera, et cetera. And so the thought was, if you had a library that was micro-printed somewhere, that you could take a small volume of a biologic fluid and apply to that. And if you knew where the individual molecules were printed, you could assign each one of those spaces for a unique protein. And if you could quantify how much protein bound at that spot, then you could essentially say, I know what protein is binding there specifically, and so now I know how much. SomaScan is a really attractive product for a lot of people because it only requires a couple of microliters of sample. So if you're doing a clinical trial and you're collecting samples and you're putting them in the -80 freezer, you cannot accumulate bottles of plasma and serum. You can't accumulate 20 half-mil aliquots for every patient because it just takes up too much room. But these types of processes just require a small volume of blood used for the entire library that you're sampling. And so when you pay for these libraries, you can pay for a 1,072 library, a 2,048 library, a 7,000 K library. Their goal is to have a library that fully penetrates the known human proteome. So let's say 25,000 gene products. The problem is it's just really expensive. If you want to do the SomaScan 3,000 library on 88 samples, you're going to pay approximately $80,000. So it is not cheap. So you've got to be clear on what your hypothesis is and what you want to do, right? So in looking at this, here's an example of an aptamer. An aptamer is a molecule that folds up into a defined three-dimensional structure. That structure binds to a specific molecule in kind of a key and lock type motif. And so if you change the molecule, you change the structure, you change the binding affinity for different things. This is a DNA aptamer. So there are DNA aptamers, there are fluorescent molecule aptamers, there are antibody-based systems as well. So the SomaScan operates by having an agarose bead that has one of these capture molecules on it. And you apply your sample to it, and it's going to bind your protein specifically. And then you apply a solution that is going to chemically biotinylate all the proteins in your sample. Then you are going to release your sample and allow it to stick by the biotin to a streptavidin magnetic bead. So that's going to allow you some measure of specificity by controlling false discovery rates because you're capturing, biotinylating, and capturing again, and then quantifying. So you have a systematic control over non-specificity here, right? There's another method that is an aptamer-based method, but it's called proximity extension assay. It's an antibody-based method. And in this antibody-based method, the idea is that this company has developed paired antibodies for every protein. And those antibodies have a DNA primer on them. And so if those two antibodies bind specifically to where they're supposed to bind, those two DNA sequences will anneal together. And so then there is a pre-amplification step and then essentially a RNA-seq step that's going to quantify the amount of the original annealed DNA primer. Okay, so the control for the false discovery rate here is the two antibodies have to bind to the appropriate epitope so that you get annealing. So if anybody in here has done Western blots before, you know, antibodies, if you expose your Western blots for too long, you get lots of bands all across your film because of non-specific binding. So you have to control for non-specific binding of antibodies, and that's how this is taking place. So these are the two concepts that I'm going to talk about. I'm going to say SomaScan or I'm going to say Olink, and this is the process. So Andrei Kolevsky is a long-term nephrologist, epidemiologist at the Joslin Clinic. And he had a really great idea back in the 90s to start collecting biospecimens on every patient that he saw. And he just simply accumulated a large biorepository of samples. He maintained them, they were his biorepository, and he followed his patients for 20 years. And through longitudinal follow-up, he could identify people that had They lost their renal function. Maybe they developed microalbuminuria. Maybe they developed macroalbuminuria. Maybe they had specific panels of antibodies. He was able to clinically characterize his populations of patients extremely well. And then what he did was essentially say, for all of these patients after 20 years, I know you. He went back to their baseline samples and started asking questions. What is different in the baseline sample? If I want to have predictive capability, I need to go back in the time machine, back when nobody has disease, and see if the molecular characteristics there are different. And so that's his approach to searching this. And this is just some data that is just addressing the fact that the age of onset of ESRD, end-stage renal disease, or CKD stage 5, is hitting in most of these patients in this population in their 40s. And within this population, over time, there's a steady increase in the number of individuals with CKD stage 5, whereas the younger individuals, the relative change in the population is not increasing. These two plots are looking at a patient set of samples from his study, where this represents the people that he's went into his freezer. So he studied all of these patients longitudinally, 12 to 14 years, identified individuals with stable renal function, and then went into his Wayback Time machine at the baseline entry. And the baseline here is when microalbuminuria was first observed. And he was able to compare these samples versus these samples for all of his studies. Right. And so through all of these studies, what he concluded was that there was a paradigm for the development of diabetic nephropathy that was largely being driven by early renal function decline, not from microalbuminuria, not from proteinuria, but from some causative factor that is causing the kidney to have reduced capacity to ultrafilter the blood. Right. Now, there are three bubbles here, and then this last one. So you have healthy patients with type 1 diabetes, and about 10% of these individuals will start losing their, well, 10% of these people will develop microalbuminuria. But you'll have a larger fraction that will begin this inexorable progression for loss of renal function. So if you have people with normal albuminuria, about 10% of them will develop, 10% will develop early progressive renal function decline. So they're going to lose more than 3.5 mils per minute per year in their renal function. And over time, this rate can vary a little bit. It can be much, much more than that. At a minimum, it's that amount, but it doesn't stop. And so 10% of these people progress and develop microalbuminuria, and then 25% of those people develop proteinuria, and about 50% of those people progress to end-stage renal disease. And there is some predisposition factor to having reduced renal function that incites more proteinuria. So the first study I want to address is one by Monica Nifkas, and this was a study that developed the idea of a kidney risk inflammatory signature. So the background hypothesis for this is the progressive loss of renal function is not due to protein in the urine. There's some other causative factor coming from the renal parenchyma, the renal stroma, a circulating cell that's inducing this change. And so they studied a cohort with type 1 diabetes out of the Joslin. They called it their discovery cohort. They had a validation cohort out of the Joslin that was comprised of type 2 diabetes, and then a replication cohort that was the Pima Indian, which is a type 2 model. And so what they used was the SomaScan approach. So for all of these patients, they took a few microliters of blood. They went through the SomaScan process, which is a chip-based process where you're applying the blood. It's binding to different locations on this chip, and then they're quantifying the amount of these reporters that are in different locations on the chip. And what they found was that in individuals that had progressive loss of renal function, they found an enrichment of these tumor necrosis factor receptor proteins. And so there were about 17 of these protein factors that were increased in the progressors, and they were associated with an increased hazard ratio, both in the discovery and the validation cohort, as well as the Pima Indian cohort. There were other proteins that were also enriched that were cytokines and chemokines, but they focused on these tumor necrosis factor receptor family of proteins. So this is, I need to move fast because I talk slow, but this is some specific details on the effect size for the increased relative abundance for the TNF receptor proteins in the progressors versus the non-progressors. This is a minus 10, this is a log 10 P. So they could have made this minus log 10, but essentially this is 10 to the negative 18.7, right? So that's such a small number, they didn't want it to have 0.00000, whatever. This is just a nice convenient way of saying it's really statistically significant. So you can see that these tumor necrosis receptor family of proteins are enriched across the board with an increased hazard ratio. So they mined the data for both the receptors as well as their ligands. And what they saw was that there was an increase in the ligands, but the increase in the ligands was not directly proportional in every case. So the receptors are having, these are circulated shed proteins that are being released from some population of cells. And conceptually, they may or may not have the ligand bound to them as they circulate, or perhaps they're binding to some other cell surface, maybe within the kidney. But this is developing the idea that there is a kidney risk score that's involving these pro-inflammatory signaling pathways. They did consider, can these molecules have any potential as a biomarker for response to treatment, right? And so this is a comparison, and I'm not making any recommendations because I don't have a medical license, right? So this is anecdotal, but this is a comparison between the drug versus placebo. And within a population of type 1 and type 2 diabetics, I don't have the numbers there because I didn't want that to muddle the data. But essentially, with losartan, with RAS inhibition, we're not seeing large fold changes, but we're seeing changes in tumor necrosis factor receptor expression. And with the JAK1/2 inhibitors, we're also seeing changes in expression. Since this is a ratio of treatment over placebo, with treatment, it went down. So the log 2-fold change would make that into a negative value. So anything that's below the line would be something that goes down with treatment. And so the red values, I believe, are in type 2 diabetics, and the gray are in type 1 diabetics. So there is some idea that quantification of these inflammatory signature panels might have the potential to be a biomarker that's predictive of future loss of renal function and is predictive of response to treatment. So a second study that came out of the Joslin was a study that used the other approach, the O-link approach, the antibody approach, because the SomaScan did not have a good reception across all labs across the nation, because nobody wants to think that you can take a microliter or two of blood and assay 1,000 or 5,000 proteins accurately. So within the Joslin kidney study, they have both type 1 and type 2 diabetics. They focused at looking at the individuals with type 1s with macroalbuminuria and type 2s with micro and macroalbuminuria. They focused on the individuals that had EGFR greater than 45. So you would put it stage 3, stage 4. And then within those individuals, we had some individuals that progressed to end-stage kidney disease and some that did not progress, right? So this is a progression, fast development. So they tried to make a distinction between this study and the other study as being this one is regarding fast development of ESKD and the other one was not fast development. But I think truly it's just the difference between they did SomaScan in the other one and they did Olink in this one. And so this is the discovery study, the validation study. And again, with this study, you have a volcano plot that's showing you enrichment in the individuals that had development of ESKD. And the red dots on the plot are things that stayed significant after Bonferroni adjustment. And you'll note that there are two proteins that they highlight. One is KIM1, which is kidney injury molecule 1. That's a hepatitis C receptor protein that's associated with tubular injury. And the other one is a tumor necrosis factor receptor family protein that was in their other factors. So they were able to simply profile those significantly differentially regulated proteins, do some cluster analysis on them, look at what other pathways that those proteins inferred beyond just tumor necrosis factor receptor signaling. And what they identified was an overlap with apoptosis and apoptotic processes. So since they were analyzing blood, they wanted to ask the question, where are these proteins coming from? Could they possibly be coming from inflammatory cells? So they isolated white blood cells from these patients. The triangles are cases, the circles are controls. They used a qRT-PCR or an RNA-seq method. And for all the proteins that they identified out of the 46, they didn't see any of them that were differentially regulated in the white blood cells. They did go back by a different method and look at the plasma, and they did see the red dots there enriched in the plasma. So the proteome is being enriched in the plasma, two different approaches, but it's not coming from the white blood cells. So then they looked at kidney cells isolated from donor kidneys, and the controls, I believe, were remnant tissue from excised tumors, and they were at least 2 centimeters away from that tumor. But they were able to do single nuclei RNA-seq analysis of the microdissected cells, microdissected tissue that was dispersed in the single cells and then into single nuclei used for RNA-seq analysis. They looked at their crisp apoptotic proteins across the different populations of cell types. So proximal tubular cells with injury, that's the PT with the VCAM1 expression, parietal epithelial cells, thin loop cells, distal collecting, you see plasma cells, intercalated cells, podocytes, endothelial cells, mesangial cells, fibroblasts, and leukocytes. And what they found was that the crisp proteome was being associated with injured proximal tubular cells. That was their conclusion. So the injured proximal tubular cells, the proteins that were being differentially regulated within the individuals with diabetes and ESRD, the few that they were able to get, I think maybe 24 total is what they had versus 10 controls. They developed this hypothesis that the injured proximal tubular cells are undergoing apoptosis, and these apoptotic cells are being released into circulation. I'm not really sure how that happens because the proximal tubular cells, when they die, they should be sloughing off into the urine space. But somehow the hypothesis is it's being picked up at the basolateral side and being pulled back through the endothelial cells into circulation. They were able to take this score, they were able to take those proteins from their Olink study, overlap it with the SomaScan study, find a subset of proteins that were in both studies, and then look at a cumulative incidence of doubling of creatinine or achieving ESRD. And they were able to show that there was, if you will, a dose dependence of this process, where the more there is of the crisp apoptotic signature in the blood, the higher the incidence you have of developing ESRD. Okay, so we've had blood and we've had blood. This study came out of University of Pennsylvania from Caitlin Sustak's lab, where essentially what they did was to look at the tissue. So they were able to microdissect out tissue from donor kidneys and use a SomaScan proteomic approach. They took the tissue and did SomaScan on this, which is a little bit more difficult. And you can see the total populations of samples that they had. They did a cluster analysis, and the cluster analysis on the differentially regulated proteome largely clustered the controls together versus the diabetic kidney disease. So they looked at a correlation of these proteomes correlating with between the patients and their estimated glomerular filtration rate versus interstitial fibrosis. And what they were able to find was about 13 proteins that overlapped as being associated with both EGFR and associated with fibrosis. That doesn't mean that they were both, all 13 were all negatively correlated with both or all positively correlated with both, but there was just a significant correlation. And so within these proteomes, they identified a strongest correlation of MMP7 with fibrosis. So this is an approach for taking clinical samples using a SomaScan approach and identifying proteomes that correlate with risk of fibrosis and risk of loss of renal function. So this was a focus on understanding MMP7, or the role of proteases within the development of diabetic kidney disease. So looking across their entire data set, they filtered out all the MMPs and the tissue inhibitors of metalloproteinases. And what they found was that MMP7 was specifically enriched for it and associated with a negative correlation coefficient with EGFR and a positive correlation coefficient for fibrosis. They looked for this process with a couple of other methods, looking at correlating RNA-seq data with SomaScan data, and the data was very consistent that MMP7 was upregulated in diabetic kidney disease. They took a similar approach as the last study to look at single-cell RNA-seq to try to identify the sources of MMP7. And what they arrived upon was that there was an enrichment in the expression of the RNA in the parietal epithelial cell as well as the thin loop region, right? So then they were able to go into a completely different data set for validation from the ERIC study and look at both plasma and tissue, looking at RNA and protein, and we're able to validate the association of MMP7 increasing with fibrosis and decreasing with the loss of renal function. So our approach is somewhat similar to these other studies in that we're looking at the degraded fragments of proteins in blood and urine. So we're looking at the end product of metalloproteinase degradation of protein, or protease degradation of proteins, maybe metalloproteinases, maybe not. So there are about 350 identified different proteases. They fall into categories of serine proteases, cysteine proteases, aspartic acid proteases, threonine proteases, and the metalloproteases. So those are the basic family of proteases out there. Oops. Right. And so I think most of you have a conventional knowledge of the role of proteases in health and disease, and you probably have a better association or affiliation with proteases and cancer, because the general concept of metastasis involves cells migrating through interstitium, and so you're needing that interstitium to be broken down so cells can migrate through there. Right, so this is a table I have on 2 pages that simply addresses specific proteases with specific types of uh, cellular processes. So with cell proliferation, we know that proteases such as MMP2, MMP3, ADAPs, cathepsins, kallikreins, these are involved in extracellular matrix remodeling, signaling from the outside into the cells, processing growth factors, and boosting proliferation. For resisting cell death, MMP7, HTRA, caspases, cathepsins, similarly, they have roles in modifying and modulating cell cycle, cell life, cell death, etc. Evading growth responses, supporting replicative immortality, angiogenesis, neovascularization, metastasis, and invasion. So our hypothesis with our study is first to perform the proteomic study, which we call a degradomic study because we're looking at the degraded protein fragments, and agnostically compare the data sets across cohorts of patients and identify those things that are different. And so once we identify those things that are different, we'll begin to explore that biology. Right? So MMPs in the kidney have links to both renal protection and to damage. So specifically, since Caitlin identified MMP7 as associated with diabetic kidney disease, I wanted to focus a little bit on the explanation for this. So MMP7 is upregulated in CKD. It's upregulated by beta-catenin activation, and it has a pro-fibrotic effect, right? So it induces EMT by degrading E-cadherins, by degrading collagen 4, and by degrading laminin. It also has an effect on the glomerular filtration process because it can mediate shedding of nephrin, one of the slit diaphragm proteins. And then lastly, in acute kidney injury, it can actually have a protective effect through shedding of the Fas ligand, right? So we have to be clear on our context. So I'll bounce through this slide pretty fast. The left side is historical data where we did degradomics with the Joslin and had some really nice discoveries and confirmation. And the right-hand side is a set of aims that I thought was straightened out better than it was. Apologies. But this is a grant that was submitted by Tina and Takas and myself. That was reviewed and scored, and we need to resubmit, and we had 3 aims in that. So we performed our study is based out of the Pittsburgh Epidemiology of Diabetes Complications study, the EDC study. Here's some information on these samples, on the population of patients being studied. It has a little bit of a negative, not a little bit, it has a criticism of being an older study where all the samples have largely been collected predate modern treatment, modern therapies. My perspective is the pathways are common to today. So in our study, in our pilot study, where we performed a degradomic analysis to identify both plasma and urine protein fragments that were associated with renal function decline, future renal function decline, we looked at paired urine and plasma samples. We sampled a subset of both male and female patients, and we only selected those individuals that had longitudinal data available for us. Our workflow is pretty simple. We take plasma, we do an acid precipitation of proteins, isolate the fragments the low molecular weight components of the plasma. We clean that up and we do a direct analysis. We do some de novo sequence assignments, and then we form our statistics. What was really impressive about this set of samples is that we identified a robust signature globally, over 22,000 different peptide fragments in the blood and in the urine. The peptidome, we saw a unique composition for both the plasma and the urine. And in some cases, we saw unique fragments for proteins in the blood as well as the urine. So we took those 22,000 fragments and then computationally imputed the proteases that were responsible for the N-termini and for the C-termini. And we looked for statistical enrichment of proteolytic activity so that we could begin to understand what proteases might have roles in the data set we're studying. So the top 10 proteases I've listed here, and if they're red, then they're statistically significantly upregulated in our data set. And out of the top 5, we have 3 of them as matrix metalloproteinases. This is for generation of peptides at the N-termini. When we look at imputation for proteases responsible for cleaving the C-termini end, out of the top 5, again, 3 are matrix metalloproteinases. So our data set for our patient population jives very well with Ketlin's from a completely different approach, that matrix metalloproteinases, and specifically the same as she saw significantly upregulated MMP7, appear to have a role in the generation of our degradome. We evaluated our proteomic components to look for things that would be coming from degradation from the matrixome. So if fibrosis plays a role in the progression of a disease, then we're going to have remodeling of the extracellular matrix. So we look specifically to see how diverse was our data set in terms of the extracellular matrix. And when you talk about the extracellular matrix, you really talk about the matrixome. The matrixome has both the structural components as well as the associated structural components. And what we saw was that our data set was very rich in collagens and the glycoproteins that are already documented to have roles in kidney disease, proteoglycans, ECM regulators, ECM affiliated proteins, and secreted factors, including a TNF receptor as well as epidermal growth factor. Right. So then what our goal was to take this data set, start looking at unique peptides, start aggregating the data and consolidating all the fragments of common to a specific gene product for something that we call the pseudoproteome, and compare that across the data sets. And then to look at integration of the plasma and the urine degradome, because the glomerulus is thought to be a size filtration with a molecular weight cutoff of about 60,000. All the fragments that we're looking at are below 10,000. So there is the potential for peptides to be ultra filtered, right? So we use a variety of different ways to look at the data. So this is a PCA plot. So principal components analysis plot. This is a clustering approach to look at your data. So when you have a lot of data sets, 22,000 data points across 16 patients, that's a little too complex to look on a graph and understand the data. So PCA will convert your dimensionalities down to a simple XY coordinate. So that's the value of a PCA plot, is it compresses your data dimensionality down to 2 dimensions. And what we can see is on this plot, these 2 plots are the same data. It's just the data points are colored differently. So here, if it's red, it's a female sample. If it's green, it's a male sample. If it's a circle, then that means that individual in the future developed ESRD. If it's a circle, they did not develop ESRD. The circle has stable renal function. The triangle, they did develop renal function decline. And what you can see is that when we consider the data from those 2 dimensions, future renal functional loss and sex, we don't see clear patterning in the data, whether we're looking at principal components 1 and 2, 1 and 3, 1 and 4, 1 and 5, etc. But when we come over here and we color according to the source of the sample, red is plasma, green is urine, and then no renal function loss and renal functional loss, what we start to see is that the data clearly has information content that is significantly different based upon the source. So when we begin to look at the data and mine the data, we're going to consider the source of the data different. We're not pulling it together. Considering all of these categories of samples, 8 different comparisons, we looked at the data from an ANOVA standpoint, and we can see that we have large amounts of significantly differentially abundant peptides associated with extracellular matrix proteins as well as plasma proteins. When we begin to look at individual components, fibrinogen B, ApoA2, ApoC3, ApoB, collagen 1A1, PIGR, and uromodulin, what you can see based upon, you can't read it, but the data are paired such that it's looking at plasma, urine, plasma, urine, plasma, urine, plasma, urine, plasma, urine. And what you see is for most of these highly abundant differences, again, we're seeing distinct differences between the source of the sample. But when we look at more advanced approaches using partial least squared discriminant analysis, what we can begin to do is pull apart the different subgroups of samples, and using a clustering approach, a heat map approach, we can begin to dig out the features that are driving the separation of those samples. So I realize that those are too small to read. I did begin to look in a pairwise fashion for differences between male urine, people who had stable renal function versus people who had early renal function decline, female urine, and the same with plasma for male and female. And by and large, female urine is not as complex and different as male urine is. We see large, more significant differences in the male urine and the male plasma than we do the female plasma. So that's somewhat not surprising in that there are known information on sexual dimorphism and the response of diabetes. So sex hormones, hemodynamics, adiponectin levels, oxidative stress response, water and electrolyte homeostasis, and risk factors. There are known sexual components to that. So instead of trying to take the individual components separately, I aggregated the data for female with renal function decline, female with stable renal function decline, male with renal function decline, male with no renal function decline, and began to pull out simply the features that were differentiating our samples based upon sex and disease. And so there are a variety of proteins over here that you can see. The one I homed in on here is epidermal growth factor. And the other protein that I homed in on was PGRMC1. It seems to differentiate our patient populations when we aggregate it all together, both yeah, I think I'm showing the same thing twice. So we looked at both, we looked at the strongest features here, and just when we compared the data, compiling sex together, compiling urine and plasma together, and looked at just the effect of EFRD, we found strong features for PGRMC1 and ApoA2. So using those two features, we asked the question, could we categorize our patient population, and could we use those features to differentiate predictably our sample sets? And PGRMC1 is able to, with a receiver operator characteristic curve, to differentiate both renal function decline and stable renal function. It's higher, more abundant. The fragments are more abundant in the renal function decline than in the stable renal function. So this protein appears to be degraded. It's either made more and has a stable degradation rate, so there's just more stable, there are more products, degradation products, or the rate of degradation is increased. ApoA2 seems to be more predictive of normal renal function. We did some statistics on aggregating things, multiple markers together to see if we had better predictive capabilities, but we're still in the process of doing that. Haven't concluded on, do we have a multi-marker panel or a single marker panel? I did dig a little bit into the epidermal growth factor domain structure to try to localize where our fragments that we were observing, wherever they were located within the protein. EGF is coming out to be a marker of healthy kidneys. Epidermal growth factor is its expression decreases with age and it decreases with kidney damage, both acute and chronic. So epidermal growth factor itself is a really rather small peptide, right? Epidermal growth factor that you use and quantify with an ELISA is a large protein that has a transmembrane domain, right? That should be pointed at this black right here. This transmembrane domain, the majority of the protein exists outside the cell. It's cleaved by a couple of proteases and released to circulate. These domains, this LDL receptor class B and this EGF-like repeat domain, are used to interact with and bind to other cell surfaces where EGF can be proteolyzed and released locally and have an effect locally. So the peptide fragment that we observed actually bridges onto the EGF itself. And so what we did was to take an ELISA, quantify EGF in both our urine and our plasma samples, look at those data for the correlation for the concentration of EGF in our samples with urine creatinine from overnight sampling. And we saw, in fact, that as we would be predicted, it has a positive correlation. When we looked at the correlation of EGF with albumin excretion, we saw a negative correlation. So that would be as predicted. When we looked at the urine protein normalized to albumin excretion rates, we saw that it was increased in the normal renal function and goes down precipitously with ESKD. Our peptide mirrored that process as well, but when we normalized the protein level to the transmembrane peptide level, what we saw was that we differentiated really the female normal renal function samples from all the other samples. So we're following up on PGRMC1 for a similar analysis. So conclusions, early renal function decline in type 1 and type 2 diabetics is driven by non-albuminuria factors. Kidney risk inflammatory signatures predict early renal function decline better than microalbuminuria or EGFR. These proteins, these CRISP proteins, are released in the plasma, possibly by renal epithelial cells undergoing apoptosis. MMPs are probably involved in the shedding of these biomarkers, such as the CRISP biomarkers or our degradomic biomarkers. And these degradomes may perform better than the intact proteins for predictive capacity. And so at 5 o'clock, I have these acknowledgments. So I'll be happy to try to answer any of your questions.
Are these do you have any comparison with other kidney diseases which also have protein?
I do no. I have used a similar approach, the degradomic approach, to look at nephrotic syndrome type samples. And we do not see the EGF, or we don't see a similar data set. And I think that's largely because those studies were performed on an older instrument that had lower sensitivity. So the data set we generated here is 4 to 5 times increased what we did previously.
So these are unique to diabetic kidney disease.
A lot of these appear to be unique so far, yes. But, you know, renal function decline and apoptosis, that's a common feature. So the goal will be to identify unique facets of this. So for us, we have both the peptide, which we can quantify, we have the parent protein, which we can quantify, and we have the proteases that are being imputed. So I think we have three different features that we're hoping to compile together as compared to just simply looking at a protein. So hopefully it's conditionally unique. Hopefully the disease condition has a specific effect on the protease expression, the protein expression, and steady state degradation. Yeah.
So you would think that glucose is what drives this, or?
I so the plasma levels, I don't know. I'm not going to say that that's the case because it would have to be glucose induction of the proteases and glucose induction of the proteins. So for the ELISA data that we had for EGF and the degradome data for EGF, I'll tell you that they were coordinatedly expressed. So in the samples that had high protein, we also had high transmembrane, high peptide. For PGRMC1, we have the opposite. We have when the PGRMC1 peptides increase, the protein is decreased. So the kind of the steady state feature of this is going to be case dependent. Yeah. Yes, sir.
That's all. So if compared to plasma or serum proteomics, is it feasible use the urine protein proteomics to do some study for diagnosis?
Yeah, so half of our data set, that the last data set
Or compare
was urine. Yep, so for each patient, we had both a urine and a blood sample collected at the same visit.
consistent, right?
Yeah,
Oh,
yeah.
okay.
We tend to like to use plasma better than serum because serum is a clotting process that requires proteins to be degraded for clotting. Whereas plasma, we try to use EDTA plasma, which EDTA sequesters the divalent cations to prevent coagulation, but that also inhibits MMPs as well. Yeah. Yes, ma'am.
Like call big, you look at this dot.
Endostatin, yeah. So we do see endostatin in some of our data sets, but I haven't looked at any of this data in terms of bioactivity. Right. Yeah,
One of
that's
the things that last few years has been quite dramatic has been the role of SGLT2 inhibitors and things like that.
Yeah.
You know, so are there any early predictors that you can use when you use those drugs? Will these change?
I have no idea.
Because those are hypothesized related more to renal hemodynamics and things like that.
Yep.
So
Yep.
this does
It'll have
seem
an
to be,
effect.
Hemodynamic
Right.
related.
Right. It's a good question. Obviously needs to be done.
Maybe they.
How it actually filters out and changes practice, I don't think it will, really. Because collaborators I work with that study pediatric kidney disease, when a adolescent shows up and has protein in the urine, first thing they do is give them corticosteroids and then ask what's wrong. So they treat first and then get a handle and then understand. SGLT2 inhibitors are so widely used now, not just for diabetic kidney disease, but just for kidney disease, that it's going to be difficult to get the funding to study this, because people are going to say, why are you studying that? Because we've got these drugs, they work. We don't need to know about that because we've already fixed the problem. Right. So, yeah. So we're,
Nothing would be like most drugs, it doesn't work in everyone. So
exactly,
whether, is it a useful marker for predicting who will work? And if I don't see a change, it says it's not gonna work.
right, right. So in some ways, it would be good if we identified common mechanisms for renal function decline that were predictive, independent of glucose or protein or drugs or infection or, well, not infection, but genetics, et cetera. If there was a predictive capacity to say, we know who's going to decline faster than age, because with age, you lose about 1% a year after you hit 40, right? And that's cumulative. And so,
Yeah, questions on the.
So she showed me how to do this.
Let's stop sharing.
Okay. Stop sharing. Okay. Girish, how close are we having novel biomarkers being used in routine diagnosis? So Girish Cotwell says, how close are you to having the novel biomarkers being used in routine diagnosis applications from plasma and urine? I would say we're probably a long way because the skill set that we have is discovery. We don't have the skill set for validation studies and clinical trials. So that's a long way. Then Ahmed says, in one slide for losartan, you have shown that altered CD55 levels. Does it mean that complement components are elevated? Have you checked for C3 or C5 or any other complement component? I believe that these are involved in acute kidney disease and will be interested to know. So I can tell you that we have specifically looked at fragments of complement in this diabetic data set. And we have, I think, north of 200 fragments of like 18 different complement proteins. So the degradome is very robust in presenting information on the complement process. We have not considered what that means in terms of diabetic kidney disease. But complement fragments are a very interesting subset of data because for that to be a complement fragment, you had cleavage by another complement protein. So that gives you some information on substrate specificity to design rationally inhibitors of that complement activation process. But again, that's not our skill set. And certainly don't have the money to do it. If I had the money to do it, it would become a skill set overnight. Thank you very much.
Thank you very much.